Good evening, guys. Good evening, Anand, Sachin. Hi, hi, hi. So, how are you all? Yep. I think everyone is fine. Everyone is like yes. Okay. So, this till 6 o'clock. We can start the class. Good evening, Samya. Good evening. Good evening, Cecily. Good evening, Ananya. Yeah. So we'll start the class. Okay. So I think you guys completed up to the transcription, right? So and we are continuing the next process. Okay. So I don't think we will complete the translation today because genetic codes need a little bit of the time. We will continue the translation tomorrow. No issue on that, right? So I think everyone of you understand what is the central dogma. So how it works, right? Central dogma like DNA to DNA, DNA to RNA, RNA to protein. The first step is the DNA to RNA. Then second step is the RNA to protein, right? Understand, right? Good evening. Yeah. So actually, you come up to the transcription, right? Up to transcription, you completed like how the process happens and what are the different enzymes involved. Can you guys tell me what is the enzyme involved in the DNA transcription? What is the enzyme involved in the DNA transcription? There is only one enzyme is involved, right? So only one enzyme. Which is that enzyme? You can answer in a short. There is no need to answer in like big lines. Okay, exactly. Correct, correct, correct. It's a RNA polymerase. Then my question is it DNA dependent polymerase or RNA dependent RNA polymerase? Which one do you guys think? Is it DNA dependent RNA polymerase or is it RNA dependent? Yeah, correct, right. So it's a DNA dependent RNA. That means it takes a DNA and it creates the RNA, right? So that's what happening. That's up to what you guys studied, I think, right? So in today's class, how this RNA is think like a message, okay? So how it will transfer into the something, okay? So I always compare this central dogma into a kingdom. Okay. So think about king as a DNA. Okay, DNA as a king. Okay, DNA as a king. It's ultimately controlling the everything, right? So then what happens? It have to send a message to a person. Think about what is it? Minister somewhere else. Minister is somewhere else. This king want to send some message to minister. Okay, regarding a war. Then what happens? If we send a message like, okay, this is the strategy and everything, then what happens? If it goes to some other's hands, then what happens? He will lose the battle, right? Because everyone can read it. They will start make a countermeasures. So what he will do it? He will make it in such a way that, okay, so it can be read by the only the minister, no one else. Okay, so he will make a message and he will send a messenger. That messenger goes to the minister and he will say that, okay, these are the things the king actually told. But actually, the messenger also don't know what he is telling. Okay, but minister understands it and he will proceed to the war and he will win. Okay, that is the same scenario you can apply here. DNA is a king. DNA is sending some message through a messenger RNA, right? The messenger RNA don't know what it's carrying, but the messenger RNA carrying that information to the ribosome. But ribosome and the ribosomal RNA know what it's actually telling, and they are making it or it's translating into the actual language, and they are proceeding with the work. Okay, that's a super kind of simple thing we can say, right? So you may also know that whatever the body process happening in our body, it's happened by the central dogma only. Okay, if I'm talking, how I'm talking because there are some control is happening. There are some protein production is happening, right? There are some DNA metabolism, glucose metabolism is happening. How the enzymes came or how that particular gene came, everything comes by the transcription translation only. Okay, so those are the total what we can say overall things, right? Hi, Hema, how are you? Yeah. So today we are discussing the things. What are the things we are discussing today? We start with the genetic codes. So what is the genetic code and how it happens, all the kind of things. Okay, and then we'll move on to the translation if the time permits. Okay, if the time permits, then we will move on to the translation process. Okay, so give me 1 second. Okay, I'll reshare the screen because there something is not working. Yeah, it's working now. Problem. Okay, before that, we will start dealing with some questions. I just waited because very few people are here. Okay, I just wanted to see more people. That's why I just waited for that. A little bit of echo, so try to arrange it because, like, I'm in a closed room. That is your school only. That's why. So we will go with the things. Okay. Really, really. Give me 1 second. It again stopped working. I don't know what happens when I come to class with a lot of like things, then it will stop working. Okay, always it always do that. This week no test. I think there is no chapter is there right for the test. I don't think any chapter is left for the test. I am from which area? Yeah. Suhas, I know all those kind of things. Okay. So think about something. We have to cancel the class or we have to use white PPT. Which one will be better, do you think? Yeah, presentation, I will do it. The better way will be the white PPT, right? So that's why. You guys have to adjust, guys. You will if you start saying simple things also, it will become very difficult for us also, right? Yeah, we'll try to move on with the things. I don't know it's working or not. The PPT, it's not moving. I will make as someone as host and I will leave and I will again rejoin. Okay, give me 1 second. Hello guys, I'm in the class only, but not in the laptop. Okay, I'm in the mobile. So let me give 2 minutes. Like it's restarting. Yep. So then we can directly start it. Suhas, can you explain me why better cancel the class? Because some others are texting, sir, don't cancel. We can take 5 minutes of time. Can you please explain? No, like why? Because like I got a complaints from your teachers that some of you are talking to the teachers in a not in a right way. Okay, so I already got like 5 6 times the same text. So this is happening. Can you please check with the student because they are texting me in the chat in a very wrong way, like all this kind of a thing. So yeah, so it's not about a single person, right? It's about a lot of people. But whatever may be, so but talking rude to a teacher is not right because I'm here. I can adjust. Like I can understand all of you because I know you guys from the very long time. But if someone comes and if you are doing something else, so they will really get like they will feel bad, right? They will feel about the things. They will feel about bad about the whole batch. So I just don't want to happen. So I told them like let me check with the people. I already got the names also. I got like 3 4 student names who always text like that, and some there should be a respect also, right? They will be say that ma'am this, ma'am that, those kind of things also I heard. So but when I'm in a class, that's not going to happen. That's for sure. Yeah. No, I can't name in a open class, but I told you right that's not going to happen when I'm in the class. That's for sure. Yeah. No. Don't take it personally. Don't take it personally. Okay, it's not about any specific person or anything. It's just a random thing. So uh, that's why I also mentioned it as a randomly because like I know and the people also know like who are those, but that doesn't matter, right? I know all of you, as I mentioned. So I know all of you because I am the one who started taking your classes, right? So you are the when you joined, I am the one who's taken classes. So I know all of you. So when anyone mention any names, I will know like who is that person and everyone. And even also like now also you guys in a contact with me. That's why also I know. Yes. So everything is working fine. Again, we are coming. There is no recovery or anything. Sure. Guys. So I'm back in the laptop. So let me share the screen. And I'm telling you, don't take it personally. Okay, don't take it personally. Yeah, anything. Yes, then I can share the screen. I can go with the class, no problem. I think everyone can see my screen properly, right? And I'm also audible. I think so. If you have any problem, please let me know. Okay. So please let me know quickly. Yep. Shall we start then? Put it in the chat box. So sometimes that happens. Okay. No problem in that. This is my third class in the day. Okay, I will move on to the things then. So first question came. This question is, yeah, I got all the things. I'm not telling that like that. Okay, but when I got a message, rather than I'm getting angry, so I will become more sad. Okay, so because I know the people, then I feel like no, so nothing like that. I will try to solve things only because don't take tension. Who all did like that already got a message from my end in the WhatsApp. Okay, so who have not got the message, there is no need to take tension. Okay, yeah. What about the NEET exam this year? This year nobody knows. Okay, even the ministers don't know. They already resigned and all the they are in their own things. So it will happen in the September first week, I think. Yeah. So, question, we'll go with the question. The figure shows that hypothetical tetra, I think this not happened with you guys, right? So this is not happened. We'll go with this question. So gene and cistron words are sometimes used synonymously because gene and cistron words are sometimes used synonymously because one cistron contains many genes, one gene contains many cistrons, one gene contains one cistron, one gene contains no cistron. I think you guys already know about cistron, muton, and recon, right? So then it will become super easy, this question, right? So first question, no, you guys don't know. If you guys don't know, I will explain. Okay. So think about it. Have you ever heard about the term cistron? Actually, sometime I think they mentioned. We don't know. Okay. If you guys don't know, then there is no this one need of launching the poll. You guys are choosing randomly. No problem. Okay, I will explain because a lot of people saying don't know. Remember, whenever we are talking about DNA, we normally talk about three different things. Okay, one is a cistron, and another one is a muton, and another one is a recon. Okay, so simple, very simple. As the name suggests, I will tell you what are these three things. Okay. The first one is a cistron. So what is the meaning of cistron? Okay, cistron simply means that the functional gene, or you can say the gene which is actually working, or you can say the functional gene. Okay, functional DNA or functional gene is called as a cistron. Okay, cistron means it's a functional. Okay, so what's the meaning of muton? Muton means the gene where mutation is happening. Okay, mutation is going on. Where mutation is happened or mutation is going on, those are called, or mutated already. Okay, those are called as muton. Okay, and the genes or the part of DNA where recombination is happening, those are called as recon. Okay, so let me tell you, think about a particular set of DNA. Think about this is the DNA. Okay, this is the part of the DNA. And some part of the DNA, what's happening? It is producing a protein. Okay, this part of the DNA, think about from here to here. Okay, I will write down like 3' to 5' and 3' to 5'. Okay, something like this. So this is producing a protein. Then I will call it as the cistron because it's functional. Functional means what? It's producing the protein. Okay, transcription is happening, translation is happening. Okay, those are called cistron. Think about here, one base pair changes happen. Okay, then it is called what? Then it's called as the muton. That means one gene change, mutation is happened in this particular base. Okay, in this particular base. Okay, think about there are two DNA like this, and during the meiosis, okay, during the meiosis process, especially in the prophase 1, okay, prophase 1, think about some part of this gene is transferring to here, some part is transferring to here. That means recombination of is happening. Then the DNA which is developed by this recombination are called as the recon. Okay, are called as the recon. So these are what you can say general terms we normally use, not that much kind of a thing, but cistron is normally asked. Okay, muton recon nobody normally uses that much regularly in your class, but cistron they normally use. Okay, so what cistron represent? A functional part or functional gene. Okay, so gene means what? Gene means what? Gene simply means functional part of DNA, right? Functional part of DNA. When you compare the cistron, the definition of the cistron and definition of the gene, it normally almost looks similar, right? Almost looks similar. Why it looks similar? Why we use cistron? Sometimes we'll use the term cistron, sometimes we'll use the term gene. They are asking why it is called like that. Okay, the reason is simple. Is it one cistron contain many genes? No, not because of this. One gene contains many cistrons? No. Remember, one gene contain cistron means gene only. No. One gene contains no cistron. Gene means functional. Cistron means functional. Right. This is also not the correct answer. Then what is the correct answer? Then correct answer is one gene contains one cistron. The gene refers to the cistron or cistron refers to the gene. That's why we normally use it as a synonym. Okay, so gene means it contains a functional gene or functional part. Okay, functional part. Okay, so that's why C option is correct, because that's why we'll use the term synonymously. Okay, so we'll move on then. So we'll directly move on to the genetic code. So yeah, so now we are discussing about the genetic code. I am telling you a story, right? I am telling you a story about the king and also the message he is sending, right? So initially what happened is like we know the DNA, okay, and we know the protein exactly, but the issue is like we don't know how the communication is happening, okay. So then we come to know that, okay, communication is happening through a mediation of mRNA, but we don't know how this mRNA, okay, how this mRNA are representing, okay. One question came, one gene contains how many muton on recon? That totally differs, right? If one gene, think about a functional gene is there, okay, one mutation site is happened, a mutation is happening, then we call it as a muton, okay, or then we call it as a recon. If the mutation not happened, then we won't call it. It's based on recombination is happening or not happening, but recon and muton always represent one functional gene. Cistron, muton, recon always represent one gene. I can't say like one cistron contain two muton. I can't say like that. If I say muton, that means complete gene is mutated. Okay, the mutation is applied to complete gene. Okay, it's about a single cistron or single gene. Okay. So RNA polymer, exactly. We know that mRNA is there, but we don't know how the message is going on. Okay. So there what happened, initially we come to know that, okay, there are 20 amino acids are there. Okay. And we also know the another one data, that's like we have 4 nucleotides. Okay. 4 nucleotides. What are the 4 nucleotides we know? We know that there are like purines and pyrimidines, those will be A, T, G, C. We know that, okay, 4 nucleotide bases are there, 20 amino acids are there. Now, we'll start thinking, then how many codon should be there, or how many nucleotide code for a single amino acids? That's the question. Okay, so George Gamow started doing his work. Okay, what he did that, he considered that if one nucleotide code for, or one monomer code for one amino acids, AA means amino acid. Okay, just remember. How many amino acids we will get? Obviously, correct, right? We will get the 4 amino acids. We will get the 4 amino acids. Understood what I am telling? So first he considered that, okay, one nucleotide we will consider as the 1 amino acid. Then 4 nucleotide, 4 amino acid. It's a fail because we have how many amino acids? 20 amino acids. How the other 16 will be coded, right? How the other 16 will be coded, right? I think, yeah. Then what he started doing, he started doing that. Think about the second one. Okay, think about the second chance. The second chance means think about two nucleotides are there. Okay, think about two nucleotides it will code for. Can you guys tell me? Think about two nucleotides are there. Then how many amino acids it can code for? Think about this probability and all these things. Think about an answer. Yes, two nucleotides forming a bunch. Okay, two nucleotides forming a bunch like AT, TA, GC, CG. Okay, AG, AC, like this. It's forming like a bunch, you can say. Amino acids it can form, but still this is also not valid, because still how many left? We're left with it 12 amino acids, right? Then what we did, he told that, so okay, I know how it's looking, that's why I'll try to avoid that thing. Okay, yeah. Yeah. So third one, they told that, okay, then consider three nucleotides. Okay, then we consider the three nucleotides. Then what are the possibilities? Okay, what are the possibilities we will get? So 16, right here? Yeah, 16. So what are the three nucleotides? If it is the three nucleotides, how many we will get? Exactly, we will get the 64. 64, three nucleotides. If we make a trimer, okay, if we make a trimer, then how many we will get? Here they were told that we will get the, I will make it little changes, codons, okay, codons, and we'll get the 64 codons. Then we come to know that, okay, so three nucleotides probably the answer, because this three nucleotide will give rise to 64 codons. Okay, this three nucleotides will give rise to 64 codons, they will thought. Okay, so then what happened? Yes. So then they told that, okay, this is the thing, three nucleotides, which will give us to 64 codons. Okay. At the same time, there are other people also started working on that. The major figure in that is the Nirenberg, Matai, and et al. Okay. In this, the prominent name is the Nirenberg. Okay. What he told is like, not only like this, he also concluded what is the amino acids which are actually coding. Okay. What are the particular amino acid which coding is actually happening. Okay, which coding is actually happening. Okay, so yeah, so it started here and it come to the this particular end. Okay, so started with the different kind of the theories. Okay, different kinds of the theory, how many may be present, how many codons will be needed. Okay, so based on that, yeah, yeah, got it. So total, he will say that one nucleotide we will get the, okay, only 4 codons we will get. This is not at all possible. 2 nucleotide we will get the 16 codons. This is also not possible. But 3 nucleotide it will give 64 codons. That means it will be possible. Okay. So that's where he concluded that, okay, 3 nucleotides are present. These 3 nucleotides code for the 64 codons. Okay. How we told that trimers or genetic code is made up of the 3 codons or 3 nucleotides by the mathematical calculation. Okay, that's also tells you that why biology also need maths, little bit of the maths, right? So this is the thing. So 20 amino acids, 4 nucleotides, and it will be like divided in this particular way. Okay. So what are these genetic codes or how it will be communicated? Okay. I will give you a simple example how the genetic code will be communicated. So think about the DNA. I think you know the DNA strands, right? Think about it will be ATGC and it will be alternative form will be TA and it will be CG, right? Think about this, the 3' to 5' and codon, and this the 5' to 3' codon. I think you guys already know which one is template and which one is not template, right? This is called as template and this is called as the non-template, right? Or you can also call it as the coding strand. Coding strand. So what happens? Transcription happening, when the transcription happening, the mRNA sits in this 3' end and it will start to move around this corner and exactly. Then what you will get? You will get a mRNA. We should write them in order wise only, sir. No, I'm just hypothetically I'm taking, right? I'm taking it hypothetically. That's why I write down all these things. Okay. Yeah, I will go slow. Sorry, what happened is like I'm in the crash course, right? That's why I become a bit faster. Okay, I will go slow. Okay, so I will explain from here. Think about it's a double-stranded DNA. Okay, in the double-stranded DNA, there are 2 strands will be there. Okay, the first strand will be 3 strand to the 5 strand. Right. So what we call it as 3 strand to 5 strand, we call it as a template because mRNA sits here and it will produce the, this particular RNA polymerase sits here and produce the mRNA, right? The other strand, which is 5' to 3' is called as the non-coding. Why it's called non-coding? It's called non-coding because, it's called coding or non-template because it won't code for the mRNA, but it looks like the mRNA. Okay. Now I write what is the mRNA sequence you will get here. It's a 5' to 3'. As you can see here, A codes for, if A is there, the complementary base pair will be on the mRNA. The complementary of base pair is the T, but it's a mRNA. So RNA, it should be U, right? For the T, complementary will be A. For the G, complementary will be C, and the C, the complementary will be G, right? So this is the mRNA you are getting. But when next what happens? Next process will be, this is the transcription process, right? So the next process will be translation process. The translation process gives you what? The translation process will gives you the complete amino acid, right? The complete amino acid chain you will get. Amino acid chain you will get. So this particular conversion mediated by a tRNA. Okay, it's mediated by a tRNA. This is the process we are learning now. Okay, now we are learning that which particular nucleotides or the codons will code for the which particular amino acids. Okay, let me go for the genetic code chart now. Okay, so as I mentioned, there are 3 nucleotide bases are there. That means as he calculated, so it will be triplet codon. Then if it's triplet codon, then only we will reach the 64 codons. Then only we will get a possible that 20 amino acids will be coded, right? So we will write it in such a way that, so you can write it like, you can remember in this particular fashion, UCAG, UCAG, then you will calculate it's like UCAG, 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 UCAG. Okay. So if you consider from this particular fashion, okay, UUU, that means U, and U, then U and C, UUA, and UU and G. Okay, exactly. 3 pair once in a mitochondria. Don't go for the mitochondria. We will go for the mitochondria later. Okay. So when you consider this 3 set of the codons, it will become a 1 codon. Okay, 3 set of the nucleotide become 1 codon. Okay. So each of this totally we have this 64 codons. Okay. In this 64 codons, it will code for how many amino acids? 20 amino acids. In this, how many code for 20 amino acids? In that, I can only say 61 code for the 20 amino acids rather than the 64. Why I am exempting 3? Because 3 are called stop codon. Okay. These are called stop codon. Okay, the stop codon do not code for any amino acids. Okay, then if you go for the biomolecule in the chemistry chapter, there what happens, there you will learn more about these amino acids. Okay, what are this, that means structure of the amino acids, what are acidic amino acids, basic amino acids, aromatic amino acids, and all those kind of the thing. Okay, but in the biology there is no need of the structure. They never ask about the structure except the three what is it, three amino acids which is present in the biomolecule chapter in the 11th. Okay, here you do you think we have to remember all the 64 codon with respect to the amino acids? No, that's also not needed, because they normally won't ask questions something like give us a codon and they will ask about which particular it will codes. Okay, but there are certain amino acids and the codons you have to remember. There is no need to remember all, I am telling you again, but you have to remember some of the codons. I will tell you what are those things. Okay, and what are the amino acids you have to remember. Okay, yeah, I will tell you. So I will tell what about the other things, but I will tell you what are the things you have to remember. Then I will go for the character of the this codons. Okay, you have to remember the first one is the methionine. Okay, you have to methionine. Remember methionine, and another one is the cysteine. Okay, can I write it? Sorry, like explain it. What are these represent? What are the amino acids? Just listen. Okay, this is the phenylalanine, leucine, isoleucine, methionine, valine, serine, proline, threonine, alanine, tyrosine, stop codons, histidine, glutamine, aspargine, lysine, aspartic acid, glutamic acid, cysteine, tryptophan, arginine, serine, arginine, and glycine. Okay. Why you have in this, you have to remember methionine and cysteine, because these are the two amino acids which have the thiol group. What are thiol group? Can anybody tell me what are thiol group? What particular group is called a thiol group? Answer quickly. Exactly. That means these are the sulfur containing amino acids. Remember, in total 20 amino acids, only 2 amino acids have the sulfur. That those will be methionine and the cysteine. Okay. So remember, whenever or wherever you think that double strand is there, okay, a disulfide bond is there, okay, disulfide bond is there, then it is the reason of either methionine and cysteine. But if you see the structure of methionine, methionine have a closed, what you can say, thiol group. Okay, closed thiol means after the sulfur, it have a CH3. Okay, that means what? It is not involved in the disulfide bond formation, right? Then which particular amino acid involved in the disulfide bond formation? Only cysteine. Remember, cysteine is the only one which is involved in the disulfide bond formation. Disulfide bond formation. When you are studying the protein, or when you are studying the antibody, or when you are studying about the insulin, you will come across with several disulfide bonds in between those proteins. Wherever disulfide bond is there, just remember it is because of the cysteine. Okay, if you ask why methionine having sulfur also do not produce disulfide bond, because methionine do not have the free thiol group. After the thiol, there is a CH3 or methyl group is there. That's why methionine won't produce any disulfide bond. Okay, and you have to remember methionine and tryptophan codons. Okay, methionine is coded by AUG, and another one is the tryptophan coded by the UGG. What's the importance of this? In the complete these codons, only the methionine and tryptophan is coded by the single codons. Okay, single codons. All the other amino acids are coded by the multiple codons. Okay, multiple codons. Only methionine and tryptophan is coded by the single codon. So that's why you have to remember the codon. Okay, you have to remember the codon. Sir, all protein contains all amino acids in some or other amount. Exactly. The protein is formed by the amino acids, right? Protein is formed by the amino acids. So these proteins may be made up of one or two kinds of the amino acids, or it may be made up of the several kinds of the amino acids. Okay, it totally differs. Okay, but all proteins contains amino acids. Okay, but amount will be different. The number, the compositions, those things will be different. Okay, remember this tryptophan you have to remember for another one thing, that is because of this have an aromatic ring on them. Okay, it's an aromatic amino acids, and it's involved in a lot of process like what you are normally mentioned, right? Other amino acid production, or in the production of the estrogen and testosterone, in all those things the tryptophan will be involved. That's why remember the tryptophan, they normally ask question about the codon of tryptophan or codon of methionine. Otherwise they simply say that aromatic amino acids or sulfur containing amino acids, like that. Okay, so there is no need to by heart all the things. Okay, but if you are interested in the kvpy, okay, then try to remember this in this fashion, like phenylalanine to valine, serine to alanine, tyrosine to glutamine, cysteine so glycine. Okay, so when if you are search for the mnemonics, there are mnemonics are there, but the mnemonics are based on acidic amino acid, basic amino acid, like that. But why I'm telling you to remember in this way, because even though they will give you a mRNA strand, if you remember in this particular fashion, then you can find out what is the amino acids it will giving rise to, right? What are the amino acids it will giving rise to? Because you know that it's UCHE, that means U means phenylalanine, GGG means glycine. Okay, that kind of the nature. Okay, previous slide. Okay, I will sum up the previous slide. Okay, so genetic code is given by two important people, George Gamow and Nirenberg Mata and et al. What Gamow told that, so they already know that there are 20 amino acids and we have the nucleotides, 4 nucleotides. What he told that, think about a monomer, think about the monomer of the nucleotide. It will code for a this one amino acids, then it will be A code for one amino acids, T code for one, G code for one, and C code for another amino acids, right? But the issue is like we have a 20 amino acids. If we consider codon as a monomer, then what happens? 4 codons are there, then we will get the 4 amino acids. That is not proper way, right? But then they think that, okay, what happens if the genetic codon is a dimer? Okay, if the genetic codon is a dimer, then what happens? We will get almost like a 16 codons that may only code for like 16 amino acids, but we have a 20 amino acids. Then we consider like 2 raised to 3, that means nucleotide or the codons are a trimer. Okay, something like this is something like this, AUG, like this. Then they come across with 64 codons, then they say that, okay, 20 amino acids, 64 codons, there are excess codons are there, but there is no problem. It is the possible way, right? That's why they thought that, okay, every genetic code is coded by this, or every amino acid coded by the 3 codons, they thought. Okay, initially they calculated it mathematically, but as the molecular biology progressed, it's confirmed that exactly for 20 amino acids 64 codons are there. Okay, 64 means 61 functional codons. Okay, so site of production of ADA in our body, which one you are talking? Which one? Okay, so we'll move on to this thing. What are the salient features of the codons? Okay, the first thing, as I mentioned, the codons are triplet. Okay, triplet means what? Every codon is made up of the 3 nucleotide bases. It is made up of the 3 nucleotide bases, right? It's made up of the 3 nucleotide bases. It may be like something A, U, or it may be U, or it may be A, right? It may be repetitive or non-repetitive, that doesn't matter, but every codon is made up of the 3 nucleotide bases. Okay, yeah. Next, they are non-ambiguous. Okay, what's the meaning of non-ambiguous? Non-ambiguous means what? Okay, non-ambiguous means they always, one particular codon always codes for the one amino acid. That means what? if you take any particular codon, okay, if you take any particular codon, it always codes for only one amino acid. It never codes for the two different amino acids. Okay, that's called, these are non-ambiguous. You will go everywhere or anywhere, one codon is always code for the one amino acid. If I consider UUU, it always code for the phenylalanine, right? Phenylalanine. It never codes for the anything else. It never codes for the anything else. Those are called non-ambiguous. Okay. You may ask, is there any exceptions are there? These exceptions are not in your syllabus. It's not so important also, but I will tell you. Okay. There is one exceptions are there. The one exception is GUG. Okay. GUG regularly code for valine. Okay. It normally codes for the, one second, chat box. Is everything amino acid chain linkage starts from the methionine? Ha, Jayant. It actually starts with the methionine only, but later it will be changed. That's called as the post-translation mechanism. Okay, post-translation mechanism. Okay, so what is the, no response from my question? What is your question? Oh, this one, aside for production in our body. So adenosine deaminase, if you are talking exactly that particular enzyme like blood plasma or deaminase enzyme, so it is secreted within the cells. Okay, it is secreted within the cell. Okay, so no, not only in the blood or anything. Okay, it's secreted different tissues of the body, different tissues of the body. Okay, so not only in the blood. Okay, okay, shall we move on? Yeah. So next thing is, so this particular GUG, okay, so it is produced in the valine. Sir, can you repeat? Which one? Which one I have to repeat? Oh, that one. Okay, okay. So ADA means adenosine diaminase. Okay, so if that is the thing, ADA, what you are mentioning, exactly, ADA will be more in the blood plasma because of the what's that? White blood cells. Okay, white blood cells. But not only in there, even ADA secretion also happens in the lymphatic system. Okay, it also happens in the lymphatic system. Okay, non-ambiguous, I will tell you. Okay, so what about non-ambiguous? Remember, what non-ambiguous tells you that one codon always codes for the one amino acids. Okay, if you take UUU, it always codes for the phenylalanine. If I take which one? You will take something one. Okay, if I take UUG, it always codes for the methionine. Okay, it never codes for the any other amino acids. Okay, but there is one exceptions are there. The exception is the GUG. GUG in the eukaryote, it always codes for the valine. Okay, eukaryote. But in the prokaryote, here is also it normally code for the valine only, but sometimes, okay, sometimes it also codes for the methionine. It also codes for the methionine. This is the only exception for the non-ambiguous of the amino acids. Simple thing, guys, non-ambiguous means one codon, one amino acid. Okay, exception is GUG, that also happens in the prokaryote. Okay, understood about the non-ambiguous? Shall I move on to the next, this one, character? The third characters are codons are degenerate and Uber hypothesis. Okay, what they will say? They will say that the codons, okay, they will say that in some codons, okay, in the some codons, this particular one amino acid, the one amino acid are coded by more than one codons. Okay, what they will say that? They will say that one amino acid is coded by the more than one codon. Okay, example, if I take, here is only, you can see, right? If I consider, which is one? Leucine, if I consider the leucine, so it is completely coded by the 6 codons, right? It is coded by the 6 codon. If I consider the glycine, it is coded by the 4 codons. Okay, if you are confused how it's happening, you can refer to this chart. That's why I include the chart here. You can refer to the chart and you can answer this. Okay, leucine, which is coded by the 6 codons, and glycine means the 4 codons. What they are saying? They are saying one codon can be this one coded by the more than one amino acids. Okay. One amino acid is coded by the more than one codons. One amino acid is coded by the more than one codons. Exactly. Okay. Cysteine 6 codons. Exactly. Okay. So there are like a lot of codons will be involved, right? So in that they will say that. So every codon have a three bases, right? Every codon, think about U, right? So they say that this is the first nucleotide, second nucleotide, and third nucleotide, right? So or simply say that it's the first base, second base, and the third base. This Crick says that Crick, you know the Crick, right? Watson and Crick, that Crick, okay, who actually discovered the DNA double helix, okay, is they that this third particular base is there, right? This third base is called as Oberlin base. Oberlin base. Oberlin base means what? The change in this particular amino acid, sometimes that doesn't matter. Sometimes it doesn't matter. Only the first two will be considered. If I show the chart again, you can, I'm not telling in all statements, okay? In this sum statement, think about here, Cu is a common, which is actually coded by the leucine, okay? The CuHs doesn't matter. So same happen with a serine, same happen with a proline, same with happen with a threone, alanine, valine, right? And glycine. So what is happening? The third base sometimes doesn't matter. Sometimes they doesn't matter. Only the based on the first two codons or based on the first two bases, the amino acid will be determined. Okay. So what is that? Amino acid will determined by first two bases. Determined by first two bases, this is, and third base is not so important, but remember only in some codons, only in some. Why I'm telling you only in some? If you consider this particular one here, look, you see actually representing phenylalanine, A and G representing the leucine, right? That means there are certain changes or value for this third codon also, right? So it only happens in some of the codon with the amino acid relationship, but this particular, that doesn't matter, right? Third one, that is called as the Obel hypothesis. That's called as the Obel hypothesis. I am repeating, the codons are degenerate means one amino acid can be coded by more than one codons. Okay, think about any amino acid except tryptophan and methionine, those are the exceptions. Except those two, all of them can be coded by the 2, 4 or 6 codons, 2, 4 or 6 codons. Okay, but the third base is the responsible for that, because sometimes the translation mechanism do not take consideration of the third base. That's why third base is also called as the Oberlin base, and this sometime will not be considered for the amino acid recognition. Okay, amino acids recognition. Okay, understood? Any doubt in this particular page, guys? Codons are triplet, that means having three bases. Okay, non-ambiguous means one codon coded by one amino acid. Degenerate means one amino acid coded by more than one codons. Okay, mean third base pair, not pair, don't call it as a pair, so call it as a base, because mRNA normally present in a single chain, right? In the DNA we call it as a pair because it's in a double strand. Here you should not call it as a pair, you should simply call it as a base. Okay, we will move on to the next slide then. So codons are contiguous without punctuation. Okay, remember, it's not continuous, it's a contiguous. Okay, what is the meaning of contiguous? Contiguous also similar meaning only. Okay, contiguous means without any stop. Okay, without any stop. Okay, so without any stop is called as the contiguous. Okay, so what they will say, okay, so it's touching this particular codons. If I write something like this, continuous, no, it's a contagious. Okay, you can also say like contagious, we will say, right? That means simply say touching. Okay, it is touching without punctuation, there is no gap. As I write down here like that only, every codon, this is the one codon, this is the one codon, this is the one codon, right? There is there any comma or gap is there? Is there any gap is there? No, there is no gap. Everything is touching one another. Okay, why it is like this? Because it's a mRNA, everything is connected to each other. So codons are, remember, it's a contiguous, that means touching. Okay, continuous also you can say it's continuous only, we can understand. Continuous also same only, meaning is same only. Continuous means there may be space also will be there, right? Space also may be there, but there is no space in between. Everything is touching. This codon is touching this codon. This codon is touching this codon. That means that is contiguous. Contiguous means touching. Every codon is touching each other. That means every codon is so continuous, means there is no gap, there is no punctuation. Okay, so why it's happening? Because it is present on mRNA, and in mRNA every nucleotide is connected to another nucleotide with a bond. Which bond it is? Phosphodiester bond. That's why these are contiguous. Okay, that's why these are contiguous without any space. Okay, next thing, these codons are universal. That means everywhere, it may be prokaryote or it may be eukaryote or it may be plant, fungi, bacteria, everywhere the same codon code for the same amino acid, amino acid. Remember, we are leaving the exceptions. Okay, there are one or two exceptions are there. There is no need to take care of those exceptions. Okay, but if you consider in a your level, that means your 11th and 12th level, we can directly say that it is universal. That means same codon codes for the same amino acid. It may be in the prokaryote, it may be in the eukaryote, it may be bacteria, fungi, animal, anywhere. Okay, so everything it's same only. Okay, next one, they have an initiator codon. For the translation happens, there should be an initiator codon. That initiator codon is AUG. Remember, always the AUG codes for the initiator codon, which actually codes for the methionine amino acid. Okay, sir, you did not tell us mean by codon before it. Chirant, I didn't understand your question. I didn't understand. Please repeat it. Okay. So initiator codon means what? Initiator codon means AUG is always a universal initiator codon. That means it always give the methionine and it always will be the first codon in the complete mRNA. It will be the first codon in the translation process. Okay. Universal, even bacteria, exactly. Even prokaryote I mentioned, right? So bacteria, remember, universal means it may be bacteria, fungi, plantae, animalia, protista. Anywhere you take, the codons are same only. The codons are same only. Even the viral mechanism, translation mechanism also, codons same only. Okay. Why did not? What is? Bro, I told you right in the first slide only. What is codon? Codon is the genetic information which is transferring for the translation, right? First slide only we discussed that, right? First slide. Yes. Yeah, and I also told a story how it happens. Yeah. So, remember, it's always AUG is the methionine, right? Then, second, there are certain codons. These are called terminator codons. Okay, terminator codons means these codons will initiator codon, which will start the process. I will write here. Start the process. Okay, start the process of translation or the coding, and terminator codons stop the process. Stop the process. Okay, there are three codons are there. Those are called as the stop codons. Those are UAA, UGA, and another one is the UAG. Okay, every one of them have their own special names. Okay, UAA is called as ocre. Okay, and UAG called as amber, and UGA calls for the opal. Okay. Okay, Hema. Okay. So these are the three terminator codons. These terminator codons are UAA, UGA, and UAG. Okay. When this particular codons will stop the translation process, will stop the process. Why it's called terminator or nonsense codon? Because it stop the protein coding process. It stops the coding process. Okay, it stops the coding process. So remember, think about the mRNA. Here, from this mRNA, we are getting the protein. Okay, we are getting the protein, and this mRNA to protein process always starts with the AUG. Okay, it's always starts with the AUG, means methionine, and it ends where these terminatory codons are present. Where these terminatory codons are present. Okay, so this is the second rule. So codons are contiguous, means without punctuation. They are universal, that means everywhere is the same, and we are in initiator codons, or there is a codon which initiate the process. Okay, and there are also terminatory codon, that means which actually stop the process of the translation. Okay, stop the process of the translation. So these are the some of the salient features of the codon. Normally they will ask any one of the salient feature. Okay, any one of the salient feature. Okay, I thought of going also with the tRNA and explaining different loops, but time not persisted because it's already 7 o'clock, right? So we can end with here because some people also have some doubts. So if you have any doubt, I will start discussing that now. Okay, so this is the circle chart, simply means, so you can easily find what I mentioned in the early chart, right? So same chart, but here represented in a circle. Okay, think about think about from here only, where we will start something here. Okay, where we will start, where is phenylalanine. Okay, so we will start with here, right? You see, it will code for the phenylalanine. UUA code for leucine, UUG code for leucine, right? Like that. So start with here, go like this, you will find all the amino acids which are coded for. Okay, which are actually coded for. Okay, so tRNA containing stop codon is lack the amino acids. tRNA do not, that's what I thought of explaining. So tRNA, there is no, when I explain the tRNA, then you will come to know that there is this UAA, UGA, UAG do not have any tRNA base. Okay, they do not have any tRNA base. That's why translation mechanism stops. Okay, so but the time restraint happened, sorry for that. Okay, so I don't know, like sometime without knowing those things will happen, laptop something issues will happen, right? And one more thing, if you have any doubt, you can ask me now. I think is this particular like I'm going slow, right? Is it okay, the slowness, is it okay? Please let me know. Okay, or do I have to speed up, or do I have to slow down a little bit more? Okay. Okay, no problem, Anisha. Yes. So we will see these steps. Okay. Remember the first codons, second, third point, first, second. Remember the first codon point is codons are triplet. Okay. That means always codons are having the three codons. Okay. Always three bases form a single codon. Okay. So it may be any three bases of the mRNA. And non-ambiguous means one codon is always code the one amino acid. One codon or three bases one codon always code for the one amino acid. And the third point is like one amino acid can be coded by more than one codon. Can be coded by the more than one codon. Okay. Sir, are UAA, UGA, UHA are the stop codons? Exactly. UAA, UGA and UHA are the stop codons. We also given a names. Okay. These names do not have any meaning. Okay. So the first game given to the Amber because of the scientist. Okay. Scientist actually given his friend name. Okay. For a, like what you can say, dedicated something. Then other two names are given in the theme, color theme. Okay, they simply give in the color theme, but this ochre, opal, amber do not have any significance. Okay. Why insulin is being extracted from the bacteria rather from the animal sources? Remember, animal sources insulin also produced in the initially, but the issue is like our insulin and animal insulin is different. So what happens? So the person who is getting insulin will normally get some reaction, allergic reactions. Okay, that's why what they will do is like they take the DNA which actually codes for insulin protein and they transported it into bacteria. So bacteria produces the insulin. It is exactly like human insulin. Okay, that's why we use the bacteria. You guys study in biotech chapter. Sir, crossover recombination ವ್ಯತ್ಯಾಸ ಹೇಳ್ಬಿಡಿ. No, there is not much of change. Such a crossover means the chromosomes or the DNA is like crossing. Okay, that will lead to the recombination. Crossing over means like this, which will lead to the recombination. Hema, what is the doubt you asked? I answered right. Yeah. So both are similar. Crossing over means DNA is crossing itself. Okay, recombination means the DNA exchange is happening. Okay, DNA exchange is happening. So to stop the process, is 3 codons are required or any of them? Any one of the codons are enough. Any one of the codons are enough. Okay, that I thought of explaining in the translation. Then we can say that a chain after terminator initiator is present. Before every functional gene, this one will be there, initiator will be there. After every functional gene, one terminator will be there. Okay. So GUG is also an example for initiator codon. Correct, Vidya. GUG is also initiator codon, but it's in a very few scenario, like in mitochondria or in some of the bacteria. Okay. So I thought of like that's an exception, not in your level. That's I thought of limiting that. Okay. You told in second point, one amino acid code for one codons, but in third for no, Manisha, don't get confused. One codon may be, one amino acid coded by the one codon. But, one codon is codes the one amino acid, but one amino acid can be coded by several codons. Understood? So, cysteine, think about something like this. UUU always codes the phenylalanine, right? But phenylalanine can be coded by two codons, UUU and UUC, right? One more question. Hi, Hema. Let me know what is the other question. What's the other question? A plasma membrane model also proposed by Watson and Crick. No. Plasma membrane model is proposed by who? Plasma membrane model proposed by who? Can you guys tell me the answer? It is not Watson and Crick. It's someone else. Plasma membrane model. So, which process does stop codon to stop? That I will explain in the translation. Okay, how it happens. Okay. Why does toxin produced by Bacillus thuringiensis not kill the bacillus? Remember, the toxins which are produced from any organism, it will never affect the same organism. They have the resistance to that, or the toxins will be not affect that because of the condition. The toxins which is secreted by the Bacillus thuringiensis only activated in the basic medium, or alkaline medium. Okay, that condition will not be satisfied in the bacillus or not satisfied in the humans. That's why the Bacillus thuringiensis toxins won't affect us or won't affect the bacteria. Okay. Singer and Nicholson, exactly. Singer and Nicholson proposed the plasma membrane model. That's important. Okay. Watson and Crick proposed the DNA model. Okay. No, sir, not getting. Hima, we will study that in the biotechnology. There you will understand. Simply remember the toxin which is produced by Bacillus thuringiensis only get activated in the alkaline medium. Okay, only get activated, not in the any other kind of the pH. Okay, only in the alkaline pH. Okay, but think about our stomach. Where our stomach alkaline pH starts? In the intestine. But in the stomach only we have acidic pH. If you eat the Bacillus thuringiensis toxins, what happens? Those toxin is again made up of some kind of protein and some kind of the molecule. Those will be broken down in our stomach, so we won't get affected. Same happens with the bacteria. Bacteria do not have alkaline medium. Because of that, what happens? Those toxins will not get activated. Okay, except in the stem borer. In the stem borer they will get the alkaline medium. So there they will get activated and it will kill the kill that stem borer. Okay, there is no need to take tension. Once the molecular is completed, we are moving to the biotechnology. There we will explain in detail about the thing with a diagram. Okay, other doubts, guys? Is there any other doubts? Is there other doubts? Any other doubts? Sorry, initial some things confused everything. Okay, so confused means like everything like struck in between, right? No, next class madam will continue. I think ma'am is having some issues, that's why she's on leave. Okay, she will be continuing. I have other classes today. I don't wanted to cancel the class, that's why I came.